Good morning to all. I am Dr. Dhondiba, Assistant Professor of Chemistry, Government Postgraduate College, Kerala, Bangalore. Today I am going to discuss about sixth session of biochemistry, that is the chemistry 8th, which belongs to carbohydrates of 6th semester BSc. In this session, I have to discuss about polysaccharides, then sources, comparative account of partial structure of polysaccharides, biological functions of polysaccharides such as starch, glycogen, cellulose, chitin, and inulin. These we have to discuss in this session one by one. Now, polysaccharides. I have already discussed in previous session the classification of the carbohydrates. The carbohydrates are classified into majorly three categories. One is monosaccharides, second one is oligosaccharides, and third one is polysaccharides. Now here, uh, I have already discussed about monosaccharides and oligosaccharides in previous session. Now, in this session, I have to discuss polysaccharides. Now, what is meant by polysaccharides? Polysaccharides are the carbohydrates of high molecular weight which yield many or more than 10 monosaccharide molecules on hydrolysis. Or we can also be defined as those carbohydrates when they dissolve in water, it produces more than 10 monosaccharide units are called as a polysaccharide. Simple definition similar to monosaccharide and oligosaccharides, you can define that is those carbohydrates which have a high molecular mass. When they dissolve in water, they produces more than 10 monosaccharide units or monosaccharide molecules on hydrolysis called that is polysaccharides. Now, the example for polysaccharides for your syllabus, first one is starch. The, the starch is the most important storage of polysaccharide in plant cell. Is a starch. Here, it is the most important storage of polysaccharide in plant cell. It contains two components. Starch having a two components, namely amylose, that is it having a molecular weight percentage is 25% and amylopectin is 75%. Therefore, there are two components of the starch present. Namely amylose, that is 25%, and amylopectin, it is 75% to the total mass of that starch. Then first one is amylose. Amylose, it consists of a linear chains. The what is the information about amylose means amylose consists of linear chains of D-glucose units. It contains a D-glucose units having a linear structure. Linear means it is a polysaccharides means more than 10 monomeric D-glucose units are combined together, joined through a alpha 1,4 glycosidic linkages. Alpha 1,4 glycosidic linkages. Its molecular mass can vary from few thousand about 15,000. 15,000 its molecular mass will be vary. It is soluble in water. It is soluble in water. It gives blue color with iodine. The iodine atoms occupies a position in the interior of a helical coil of glucose unit that is formed when amylose is suspended in water. Therefore, this is nothing but here amylose it is consist of there is a number of there is a more than 10 number of D glucose units and they joined by alpha 1,4 glycosidic linkage to each other and it became and its molecular mass will be vary from few thousand to 1,50,000 actually. Around there is about few thousand, about 1,000, that is 15,000. It is soluble in water. The amylose is soluble in water, it gives blue color with iodine. When we are adding the water solution of amylose, it gives that is a turns blue. Iodine is a reddish brown color, but when it is added to the amylose, it turns that is the red color into blue color. This indicates it is contains a amylose. The iodine atoms occupy a position in the interior of helical coils of glucose units. It formed when amylose is suspended in water, because when we are adding that is water, the alpha helical coil will be there in the glucose units. The iodine will be it is attached to the interfacial alpha helical coil, that is alpha helical coil glucose unit, and it gives that is a blue color. This is about that is a amylose. Now coming to that is a Howard structure of amylose. Now here, I as already told now, it is contains a alpha D glucose. That is a D glucose units. D glucose unit, usually alpha D glucose unit will be there here, because it is alpha 1,4 linkage means it is alpha D glucose unit. And here, this is the H here OH is there, and this is the same, this is the D glucose. When it is joined together, this one and this one, here numbering is takes place from here 1, this is the 2, 3, 4, this is 5 and 6. This is the linkage will be takes place linear structure. Linear means straight line structure. There is a no branch structure. The one over the other, one to end, it is joining together, and to get a straight line structure, that is called as a linear structure. Therefore, here this one position of the hydroxyl group present, that is secondary alcoholic group is present out of one carbon atom, join with that is the hydroxy group or alcoholic secondary alcoholic group present at the four position of the other molecule, and deeming that a water molecule is liberated and a oxygen will be there. It is like a ether group will be there, and this is called as a alpha, that is a glycosidic linkage. Glycosidic linkage will be takes place, and because of it, these molecules are alpha D glucose, therefore the linkage will be takes place at one position and four position of other molecule. Therefore, it is a alpha 1,4 linkage of amylose. And here the N number will be there because it is a polysaccharides. The minimum the having the condensation of these alpha D glucose is more than 10. 11 it will be there, 12 it may be there, many numbers. The molecular mass will be vary around, that is a few thousand to 1,50,000 around that. That will be a vary from this, and therefore we say that is a it is a N. N value depends on the individual, that is a molecular mass, and hence it is called as a polysaccharides. Now next come there is a amylopectin. This is about that is a amylose. Amylose contains that is a alpha D glucose units, and its molecular mass will be vary from few thousand, and it contains alpha D linkase. Alpha 1,4 linkage is very essential, and it gives a blue color with iodine solution. It indicates as a that is a amylose is present. Why it is amylose is present means, suppose we are testing the iodine solution by adding iodine solution to the water solution of amylose, it gives that is a blue color. This indicates as a the sample it is a polysaccharides is nothing but amylose, and this is the straight line linear structure, and these are the few features of that is a amylose. Now come to the amylopectin. The another component of starch is nothing but amylopectin, and this is the usually there is 75% of total mass will be a present. Suppose the molecular mass is the 100 gram, then it will be a 75 gram is nothing but it is the amylopectin, and 25 gram will be of that is the amylose. These are the idea will be there here. Then amylopectin it is a branch. Now it is the amylose is nothing but straight line linear structure, but amylopectin is a branched polysaccharides. In this molecule, The short chains of the glucose units. Here again glucose units are will be there. This is the alpha glucose units. Only alpha D glucose units will be present in the amylopectin also. Similar to that is the amylose. Okay. Therefore, in this is the branch structure. In this molecule, the short chain of the glucose units linked with a alpha 4, that is alpha 1 4 linkage, joined together to each other through a 1 1 6 linkage. Therefore, two linkage will be there. One is branch, again straight line. One will be a branch, it makes the branch. Another one is small, that is a straight line. Therefore, there are two linkages will be there. One is linear linkage, another one is branch linkage. It will be takes, there is a 1 4 linkage, alpha 1 4 linkage, and alpha 1 6 linkage with each other. The molecular mass of the amylopectin is high up to 1 million. Molecular mass will be a high up to 1 million, whereas amylopectin, there is amylose having a around there is a few thousand to up to there is a 1,50,000. Not there is a 15,000, but it should be a 1,50,000 up to, but it having a high molecular mass up to 1 million. The branching occurs every at 24 to 30 residues. When it is joining, that is a 20 to 30 the molecules linearly, then it will be a take, that is a one branch. Again the branch will be a goes on developing by condensing, that is 24 to 30 molecules. Again it will be a breaking the branch. Therefore it gives, due to it gives, that is a red color. Now iodine, no, when iodine is added to the amylopectin, it gives a red color. It gives the red color with iodine. Okay. Therefore, we say that is a what is the difference between how it is identified whether amylose or amylopectin means when taking, that is the iodine solution. Suppose added to the iodine solution to the amylose, it gives that is blue color, whereas amylopectin it gives that is a red color. Whereas amylose is nothing but it is a linear structure 1 4 alpha D linkage, but here two linkage will be there amylopectin, in which it is a branch structure. One is linear 1 4 and another one is that is a 1 6 linkage. Then coming to that is a Harvard structure of that is a amylopectin. Now here this is the 1 4 linkage is it is nothing but a linear structure like a amylopectin. Amylopectin, first it is say that is just like a, this is the amylopectin, no, that is a component of amylopectin. It is a takes place. Here same there is a alpha D glucose unit will be takes place. Here alpha D glucose unit will be takes place, and there is a alpha 1 4 linkage will be takes place. After there is a 25 to after 20 to 24 to 30 residue. After joining here, after joining these are the 24 to 30 glucose units. Then will be another linkage will be takes place. That is a 1 6 alpha 1 6 linkage. One here, and this is the another one is 6. Here linkage, that is alpha 1 6 glycosidic linkage will be takes place. Alpha 1 6 linkage will be takes place. Again then straight line will be takes place, that is up to 24 to 30. Again that is branching is takes place. Such type of the branching of that is a structure, and it's here n, it is because it is a polymer, it is number n number of molecules, and n is it is used, that is the total molecular mass. Suppose we are taken as a 10, then you have to count, that is a 10 molecules. The formula will be 10, that taking 10 number into then we will get that is a molecular mass. Therefore it gives that is a molecular mass of that particular amylopectin. Therefore n is nothing but it is a value, and it will be a value up to few thousand to that is a 1 million also. And this is about that is a amylopectin. Now coming to that is a second, that is a polysaccharide is nothing but glycogen. The glycogen, it is again branched chain polymer similar to the amylopectin. Means similarly there, no, there is a 1 6 amylopectin, whatever maybe there are branched structure, again that is 1 4 and 1 6 alpha D linkage will be there. Similar to the structure of glycogen and the similar structure of that is amylopectin. And here glycogen also contains that is the alpha D glucose units. Okay, but here the branching is different. Therefore, the however structure will be a similar, but branching will be a more extensive than in amylopectin. Branching occurs at every 8 to 10 glucose unit. Every 8 to 10 glucose units. The difference between that is the amylopectin and glycogen is simple. They are 24 to 30. After 24 to 30 molecules join together, 1,4 linkage, alpha 1,4 linkage, linear structure, then it will be take there is a branching by 1,6. But here glycogen similarly takes place, but branching is more here. Every 8 to 10 glucose units, after that is joining, that is 8 to 10 glycol, that is a glucose units, there will be a branching will be takes place at 1,6. Then again 8 to 10, then branching will be takes place. Again 8 to 10, then branching will be takes place. There in the amylopectin, first one is they join, that is a 24 to 30, then branching will be takes place. Again that is at 24 to 30, after joining, then branching will be takes place. Therefore, here more number of branching will be takes place in glycogen compared to in amylopectin. Therefore, the Haworth structure was supposed, because these are the polymers, no, the Haworth structure will be similar to that is a amylopectin. I have written that is same structure. There is no change, but here 8 to 10 glycosidic, that is a residue, glucose residue, will be a branching takes place. There 24 to 30 glucose units are, that is a, it is a branching will be takes place. Therefore, we say that is how the structure of that amylopectin and amylose, that is amylopectin and glycogen are, that is the same, there is no change. Here 1,4 alpha D linkage will take place. Again, after there is a branching, after there is joining 10 to 8 to 10, then a glycosidic linkage of, that is a 1,6 linkage will take place. Again, there is a 8 to 10, there is a monomeric units are joined together. Then again branching will be takes place, and here N will be take place because it is a polysaccharide polymer, and the N is depends on, that is the number of molecular mass. The total number, suppose we are taken, there is a 30 or 40 or 100 molecules are joined, then you have to multiply, there is a molecular mass, and then getting there is a molecular mass. Therefore, this is about, that is a glycogen. Then the biological importance of glycogen, it is the main storage of polysaccharide of animals. It is the main storage polysaccharide of animal cells. Means the animal, no, they are their energy will be kept in glycogen. Second one is it is abundant in liver and also in skeletal muscle. Therefore, it is more rich in liver. The glycogen is rich in liver and that is a skeletal muscles. Then third one is it is a reserve food for, that is a energy of animals. Reserve food. When it is energy is not supplied, then glycogen undergo a glycolysis to produce, that is a again energy, and that energy will be stored, be given to the day-to-day activity for the animal. Therefore, without taking the food, the glycogen will be served the food for in the fasting. Next fourth one is it is undergo a hydrolysis enzymatically to yield glucose, which is readily oxidized to provide the energy. Oxidized to provide the energy. Therefore, I say that is here it is the energy stored, and when it is starving or it is not taken the food, then that time the this glycogen undergo a enzymatically by using a enzyme catalyst, it is undergo hydrolysis and to yield, that is a glucose unit, and glucose is further oxidized to provide, that is a energy. And this is the biological importance or role of glycogen in biological systems. Now coming, there is a third, there is example for polysaccharide is nothing but cellulose. It is a linear structure again, linear structure consisting of 10,000 to 15,000 D-glucose units. Here, this is cellulose is nothing but it is short like a linear structure similar to this is the amylopectin. This is the similar to amylopectin. Sorry, it is similar to amylose, not amylopectin. Amylopectin is a branched one, but it is a linear structure and it is similar to that is amylose. And it having a molecular mass around that is the unit should be a unit consist of this unit should be consist of 10,000 to 15,000 D glucose units will be present. 15, that is suppose one glucose unit is nothing but the molecular mass of the glucose is around that is 180 around, no, suppose C6H12 O6 means after calculating I think it will be getting that is 180 into this much 10,000 that is 10,000 means this like again there is 15,000 we are multiplying whatever maybe we will be getting, that will be molecular mass of the cellulose will be getting. Therefore this is again similar to that is the amylose. The cellulose is a linear polymer consisting of that is a 15 to n is equal to 10,000 to 15,000 glucose units. In this respect, it resembles amylose. It is given here. I have already explained. In cellulose, the glucose residues are linked by a beta-1,4 linkage. Beta-1,4 glycosidic bonds means here in the amylose, no, amylose contain that is the alpha linkage, no. Alpha linkage means alpha D glucose will be present. Whereas in cellulose, no, it is due to having a beta linkage means it contains a beta D glucose. Beta D glucose. Cellulose contains beta D glucose and it having a 1,4 linkage similar to that is a amylose and it is a linear structure. A glycosidic linkage will be takes place and it is highly resistant to hydrolysis. It is insoluble in water actually. It will not be given that is a water soluble by acids. It is highly resistant to hydrolysis by acids. Hydrolysis by strong acids resulting in the glucose as a final product. Suppose the dilute oxidizing agent. Suppose we are using that is dilute acids, the cellulose cannot be undergo a hydrolysis, but we are adding the strong acids, then it is undergo a hydrolysis and it produces the final product. It is monomeric unit like that is a glucose. It is a final product, and this is about that is cellulose. Now coming to there is a how the structure of cellulose. Here this is the same I have already told. This is the beta D glucose, no? Beta, and that is the beta D glucose units. Present means beta up here. This is the OH group should be up here at one position above the plane, whereas in alpha it is below the plane. Therefore here and this should be a 1,4 means that is similar. This is the only one difference, that is alpha and beta means one position. One is OH group is up means it is beta and below means that is alpha. Okay, clear? Therefore, it is the link will be takes place by using a this is the form and this will be takes place like that, and this is the linear structure will be takes place and it having a high molecular mass and n will be takes place here around that is the 10,000 to 15,000, and this is the linear, that is how the structure of that is cellulose. Now coming to that is the biological importance of the cellulose. It is found in the cell wall of the plants. It is found in the cell wall of plants. It is the most abundant organic compound in nature. It is first there is importance is it is found in the cell wall of the plants. Means it is a cellulose usually it is a kitchen of that is plant actually, no, that is a kitchen house of that is a plant. Therefore, it is found in the cell wall of the plants and it is the most abundant organic compound in nature. It is a flax and cotton contains flaxseed and cotton contains up to that is a 99% of cellulose, while wood contains that is a 41 to 53% cellulose, and rest of the other will be different. that is starch, whatever may be there. But cellulose, it is nothing but pure cotton. Pure cotton, or that is a flax. Flax, that is a, it contains, that is a 99% of cellulose, and while wood contains, that is a 41 to 53% of the cellulose. And fourth one is cellulose is also not acted upon by amylases present in the digestive tract of humans and most of other animals, higher animals. Therefore, we say that is a cellulose, no, cellulose contains plant. Therefore, animals, no, some animals cannot be digested. That is a cellulose, and because of amylase, no, amylase, that is a, it cannot be produces and it cannot be get, that is a hydrolysis. It cannot be break down. The only vertebrates able to cellulose as a food for cattle and other, that is ruminant animals. These are the cattle, whatever may be. They are the eat, that is grass. No, grass contains of the rich of the cellulose, and there contains, that is the amylase, and that will be undergo digestion. The extra that is the stomachs of these animals contains bacteria and secretes cellulose, that is alpha D glucosides, and that catalyzes the hydrolysis of cellulose to form, that is glucose. Glucose, some, that is animal, there some, that is plants, no, they can be bacteria that eat, that is the plants. Some that is insects, no, eat the plants, that is leaves, whatever may be. And due to having it contains, that is a cellulase. Cellulase, that is a enzymes, and that digest the cellulose, and it break down into a glucose in the form of energy. This is about, that is cellulose. And fifth one is snails. Snails, that is snails, termites, and wood, rot, fungi, or some other, that is species. These are the species which are secretes cellulose and metabolize cellulose. Means they can be, because these microorganisms secretes the, or that is produces cellulose, and it cellulase, actually cellulase, and it metabolizes the cellulose, means it digests the cellulose. But whereas animals, that is the human beings, they cannot be digest, that is the grass. Therefore, the grass cannot be eaten by, that is a say, that is a human being, because cellulase, it cannot be produced in the metabolic activity. Then it is about, that is a cellulose. Now coming to, that is a another fourth example for, that is a polysaccharide. It is nothing but chitin. Chitin. It is the linear homopolysaccharide. Homopolysaccharide composed of N acetyl glucosamine residues. Chitin, I already told, that is a, while that is the derivative of amino acetyl and amino derivative of monosaccharides, no, it contains, that is N acetyl glucosamine, no, that is amino derivative and acetyl derivative of monosaccharides. It is nothing but it contains, that is a chitin. Therefore, it is the linear homopolysaccharide. Polysaccharide. Homopolysaccharide means single molecules is repeatedly joined together and to form a high number of large or huge molecular mass of the compound, that is called as a homopolymer. And it is a linear structure in which the beta 1,4 linkage will be takes place. The chemical difference from the cellulose is the replacement of the hydroxyl group at C2 position with the acetylated amino acid. This is the just difference, because there is a beta 1,4 linkage. Cellulose having also, I told, no, cellulose having a one beta 1,4 linkage. Cellulose having a beta 1,4 linkage, whereas in chitin also having a beta 1,4 linkage. But in this one, that is also linear, this is also linear, but in this it is acetylated amino acid, means there is a OH group at there is 2 position. This as N-acetyl group will be there. N-acetyl group will be there in this, and this will be a structure will be. This is the 90% of the structure will be how the structure is similar to that is the cellulose. Only difference is nothing but here the OH group is there at 2 position. No? That will be replaced by that is a NHCOCS3. Now this is about there is a there is a comparison with there is a structure with chitin with cellulose and how structure just I have told this is the 90% is similar, only one there is the difference is NHCOCS3 will be a difference in chitin. Now coming to that is the biological importance of chitin. It is the principal component of there is a hard exoskeletons of nearly a million species of arthropods, insects, lobsters, and crabs. These are the main components of that is a extraskeletal, exoskeleton. Exo means outer, skeletons means there is a cover, there is a thin, there is a covering, or there is protective, there is a coverage of these species contains that is a chitin. Contains that is a chitin. And hence, therefore, we say that is a it is the principal component of that is a hard exoskeletons of nearly a million species contain that is a this chitin, and these are the give that is a structure. This is about that is a biological importance of chitin. Next, fifth one is inulin. Fifth, that is an example of polysaccharide is nothing but inulin, but not insulin. Okay, inulin. It is a linear homopolysaccharide composed of 2 fructose residues. Therefore, it is similar to this is the above. It is a linear homopolysaccharide composed of 2 fructose residues. That is a 2 fructose residues in 1,6 alpha 1,6 linkage, and it is a non-reducing sugar. The inulin is a non-reducing sugar. Okay, clear? Therefore, the linkage will be there. That is a alpha 1,6 linkage. It should be a fructose one. Here, other there is cellulose having a glucose only. That is a alpha D glucose or beta D glucose. But inulin contains that is a it is a exclusively for that is a fructose. That is a fructose will be there, and the how the structure of this it is a non-reducing sugar here because of linkage 1,6 linkage will be there. Therefore, the group will be a not free for that is a reduction. Hence, it is a non-reducing sugar, and it is a linear homopolysaccharide. Homo means the single unit that is a fructose is joined together and to form a long chain structure. This is the linear structure having a inulin. Now coming to that is a how the structure of that is a inulin. Now I can write, I have written this like this is called as a fructose. Fructose is nothing but actually this is the H will be there here. H, this is the CH2OH, CH2OH, then HOH, then HOH, then H, CH2OH. This like structure, this is the nothing but fructose unit. This is the fructose unit. When this fructose unit is joined with, polymerized with, other there is a group, other there is a same group, because homo means same group is repeated joined together at 1,6. That is a at 1,6 position. That is a 1,6 position. Here you take there is a first one is here 1, and actually this will be a text B here. Actually 1,6 linkage means that is same here usually. Therefore here and it is joining together, and it form that is glycosidic linkage. It taken n times, and this is the how the structure of that is a inulin. Now coming there is a biological importance of inulin. It occurs in diarrhea. It is usually this inulin is present in plants. That is like a flowers, whatever maybe. That is roots, whatever like that. Diarrhea, dandelions, garlic, onions. etc. as a storage polysaccharides. This inulin is occurred in these plants as a polysaccharides. It is a polymer of 30 to 40 fructose units. It is a polymer of 30 to 40 fructose units, and it is also called as a fructosal with beta 2,1 linkage. Beta 2,1 linkage. Beta 2,1 linkage. And this is about there is a biological importance of inulin. Therefore, so far I have explained about polysaccharides in source, comparative account of partial, comparative account of that is polysaccharides of particular examples, partial structure and their biological structure of starch, glycogen, cellulose, chitin and inulin. I hope you have understand. Any doubt you put in comment box, I will answer them. Thank you.